Well, hi there, Mr. Bleeker here. This will be our third lesson in the DNA series. This one will focus on translation, turning an mRNA message into protein. If you need to review the process where that message was created, go back and review the transcription lesson, which would be the second in the installment. Okay, well, we'll get going here. And where we last left off was mRNA leaving the nucleus. So your mRNA molecule, situated here, is a single-stranded molecule. It's a copy of a gene or a series of genes, depending on how this is put together, that leaves the nucleus and goes to the ribosome to be the template for the construction of proteins. You can think of it as a blueprint, but a section of a blueprint of DNA. This is where it gets interesting. First thing you need to know what you're looking at here, if I zoom in over here, is where we are. Right now, what we're at is at the ribosomal site. So that's the large ribosomal half there, and the small ribosomal subunit is down here. So you can think of the ribosome as a protein generating machine. And the mRNA is going to thread through this thing. At times, I kind of look at this almost like it's one of those little top toys where you insert the key and you pull, and the top goes spinning off. It's kind of interesting. It's a similar analogy, except as we insert mRNA in this case, what we're doing is we're inserting something, and the ribosomal subunit, both of them, are going to act more or less as a stamping mechanism. The stamping that will happen is based on the code on the mRNA. So we'll stamp in amino acids, but only in the correct sequence. You notice these stamps have these, let me do a little more zooming here. These stamps right here, let's see, there we go. If this is the stamp right here, and there's another one here, this stamp is transferring in an amino acid. So this stamp is also made up of RNA, and we call it transfer RNA. And if you look at the bottom, you will notice sort of an interesting feature here. Sort of a three-footed stamp. And if you look below it, let's switch the color here. Oh, I have to accept that color. And if you look below it on the mRNA molecule, there's a corresponding code. It's a triplet code. It's read 3 sections at a time. Now we'll define those sections as we go forward. The stamp has to match those 3 sections and transfer in its amino acid, which is here. Let's zoom out a little bit. There we go. So your mRNA forms the code upon which the amino acid is going to be placed. So let's call the mRNA the sequence of codons. And the transfer RNA, which matches it, will be the matching sequence. And this is known as an anticodon. So the tRNA has to match the mRNA. We'll get into this in a lot more detail as we move forward here. This portion of the slide shows the codon and the anticodon matching. It's important that you understand that there's 2 sites as a growing polypeptide is formed. You have this site right here, which is the active site, where the amino acids are being slotted in. This is where the stamping is happening. And beside it, you have what's called the, you could think of it as the peptide site, where the peptide is growing. And if you look in this case, this will, as the molecule is forming, here's your mRNA. So as the mRNA is read in this direction, the next codon will be read, and the peptide site is here. So the active site and the P site, if you will, move along the molecule as the molecule forms. And by molecule, we're talking about a growing peptide right here. So that's your P site and that's your A site on your mRNA. Let's stop for a moment, and I want to show you what this looks like in action. So there we go. So when we look at translation, here's your mRNA sequence here. And of course it is read, as stated before, from the 5' to 3' end. Now the ribosomal subunits will attach. There's special sort of attachment initiators. But for now, as the ribosomal subunits fit into place, the transfer molecule will bring in its first amino acid. Now that first amino acid is invariably, almost always, methionine. There's lots of amino acids that we'll talk about. As a subunit comes together, let's just zoom in on that, you might notice the bonding here. Adenine, uracil, guanine. So if that is the first code or the first codon, then the matching anticodon would be here. So uracil takes the place of thymine. Adenine will match against this uracil, and cytosine will match against this guanine. Okay. The sequence of amino acids, the whole nature of the protein that is going to form, is based upon the codon that you see down here. So now it's read, this is interesting. DNA is a 4-letter alphabet, A, G, C, T. Okay, I get that. But I remember saying back a couple lectures ago that it's a 3-letter alphabet, or sorry, a 4-letter alphabet that makes 3-letter words. And by 3-letter words, what I meant was that the genetic code will be read a codon at a time, or 3 nucleotides in this case, like this. So you'll see this 3-letter matching as we go along, all the way down. And that becomes really important when we talk about mutations. What happens if something happens to one of these codes in DNA? What would that do to the protein? Don't worry, that's coming. So press play here. Now we've methionine. Once the stamping process has happened, at the primary site, you get the formation of peptide bonds. So think of it this way. The stamp has delivered its amino acid, and it's done. Now, it will go back into circulation in the body, and you consume proteins all the time. As your digestive system breaks them down, there's methionine floating around in circulation, which will more or less recharge this transfer RNA molecule. So it won't stay empty for long. It'll fill its gas tank. So it flies off. It's gone for a recharge. And in comes the next code. So this will be for phenylalanine. And I'll show you in just a moment how you can predict what amino acid is coming in. But for now, suffice it to say, let's just look at the stamping process. This becomes the P site where the peptide's growing, and this becomes the active site where the arrival happens. You can think of it almost as an airport. And as we move along, you get a new active site each time. So we bring in the next amino acid. This anticodon, CCG, matches against the codon on mRNA, GGC. And it carries on. I'll just kind of until it hits something called a stop codon. Now, there can be a position in the genetic message which says this is the termination point. The protein stops here. And that's really important because if you miss the termination point, you may add additional amino acids and you could get the protein all wrong. And that can be, when mutations happen in nature, that's one of the things we'll be looking at. So you hit the release factor. The release factor will go into position at the stop codon. And there's special stop codon messages. In this case, you saw AAU on the mRNA subunit. Okay, so we'll look at our codes in just a moment. Taking this a little bit further, this is just me backing up a little bit, showing it again. Here's the mRNA strand. There's our ribosome. Pool of amino acids, of course. And I really like this animation because it zoomed right in on it. I'm being a little redundant, but I wanted to show you again. So there's the codons. So each of these represents an amino acid. So it's a 3-letter word. Now it's dictated by mRNA. Okay, so we'll be looking at a master table of mRNA codes to know what the protein sequence will be. There's your start codon. And I really like this animation because it shows what transfer RNA is. Transfer RNA almost looks like this little guy holding his hands out to the side. Sometimes it's been called a cloverleaf. The big secret about transfer RNA is that transfer RNA is a kind of RNA. It's actually single-stranded. And it's designed in this way so that its anticodon matches against mRNA codons. And it's also designed specifically only to bind with certain amino acids. So these transfer RNA molecules are a little different. Depending on their anticodon, they'll only carry a specific amino acid. Okay, so there's 20 amino acids that are going to pair up with these codons. Nice visual of the transfer RNA molecule. You have genes to produce these in your DNA. It's pretty amazing. And we just watched the stamping process. A going with U, U with A, G with C. Good. So more or less follows the rules of that we see with purines and pyrimidines, the way they bond. And that's nice because you get to see that covalent peptide bond form. And it carries on down the site. Here comes one into the active site, and the bonding happens with the primary, and so on and so forth. Until you hit what's called the stop codon. There's some famous ones. This one's UA. I always like to refer to this guy. There's also UGA. I call it the Cro-Magnon stop codon. When that's there, when you hit that, that's where that release factor kicks in, and it's termination of the protein or polypeptide production. And there you go. So guess what you have? In biochemistry, we looked at this and we said, geez, what is that? And I said, well, think of a necklace of amino acids, almost like a bead necklace. What have you got? You've got a primary protein or a first-degree protein. Of course, it will fold up into different three-dimensional shapes. So you begin to get, you can get your alpha helixes here. As this carries on, you'll get your beta-pleated sheets. That's the secondary ones. If you look at it and you zoom out, this is what you'll see. They refer to this as a polysome, where many ribosomes are attaching and producing their polypeptides. You need the ribosomes to really attack mRNA sequences so that you can get the production of proteins. Hemoglobin, for example, is huge. So you have many mRNA strands and you have many ribosomes coming to meet those strands. The more polysomes you have, the more productive you are. So this is just a really great animation. I'll be adding this one to the course site. Okay, let's see here. I'm going to go back to our notes for just one moment. And we'll come back to those animations. Those are really good. There we are. And it's amazing because we haven't gotten off this slide yet, but that's all right. So coming in, when the transfer molecule comes in and basically delivers its payload, it has to match the mRNA. So the anticodon has to match the codon. And that's pretty important. So what we're really really this goes back to if you look at each of the codons, if each of the codons is calling for a specific amino acid, it's like each codon is a word, and as we look at all the codons, we're sort of forming this sort of genetic sentence of what a protein should look like. And I like that analogy. It goes back to DNA. Remember, your DNA was read to make the mRNA in the first place. So let's look at some of these codons and let's see how we read more or less sort of an mRNA genetic table. So if the codon that is read is GCA, that calls for alanine. And AGA, again, let's say this, let's make a point of this. This is the mRNA codon, which is going to result in the delivery of a certain amino acid. Eventually, you'll get to stop codons, UGA or UA, which would be an all stop as far as construction goes. Now, how do we read that? Just 1 second here. I've got a special sort of three-way table here for you. Now, a three-way table, if I'll just pick one here. Let's say that our codon that's requested is going to be our mRNA codon is going to be GCU. So if you looked at the mRNA and you saw that, you'd be looking and wondering, well, which relative amino acid would that be? Okay, so let's get a nice bright sort of orange here. I think that should work, just so much that we can see through it. Okay, so this is called first position. So you look at your first position and you say, okay, it's the first one is G. Then you look at second position, and I'll bring this down here so you can see it. Second position is along the top, C, and then you go to third position and you go to U. The intersection of G, C, and U, so G is this whole thing here, C here, and U. So come over and I'll use a sort of a dotted structure. GCU intersects at this point. GCU. There you go. There we go. And that's the intersection. So what does that call for? That calls for alanine. Okay, so now you know what the amino acid is. I can erase that. Let's pick a different one. Okay, let's pick C. There you go. CGU. Well, what might that be? So C is here. There we go. This time I'll just use a pen. C is here. G, a little devil, it's in my way. G is in this column right here. This is the G. This is the G column here. You can see that it's the last one at the top. So I'm sorry, but my tool gets in the way for this. C, G, and U. So I'll just CGU, C, G, and U. So there's U. This is column C right here. Or sorry, there's your CG. You're in column G. There we go. Would have been easier if I could see it. And it looks like that. So that's the C to the G to the U, and here it is. Okay. There's an interesting three-way table that you can get sometimes to help you read these. This one's a circular version of it. It's just another way of wrapping it. I can make this available for you. You can put it on the test. You would look at the first nucleotide in the codon. So if we were looking at C, and then we looked at G, and then we looked at U, and what do we get? We get arginine. It's a neat way of reading the table, because if you go back here, you'll see that you get arginine right there. The tables are equivalent. This is just a three-way table, but the circular table is quite useful when it comes to reading because you just read from the inside out. Whichever way you'd like to interpret the graphic, it's kind of up to you. But they're both very, very useful. For example, if you think of the stop codon, one was UA, you find UA right here. U to the A to the A. So the different combinations you could read, you could have UAA as an mRNA message. You could have UAG. And you'll notice there's several stop codon possibilities in here. You could have UGA. There's that combination, UGA, which is also stop. And UGG, so these are all stop codons. You have to stop this protein production point at different, using basically 4 combinations. Now, if you think about DNA, speaking of combinations, and you say, how many combinations are there? Well, think about what you have at your disposal. You have 4, there you go. You have A, C, G, and if we're talking about mRNA, we'll talk U there. So you have 4 nucleotide choices that can be arranged into 3-letter words. If you look at the number of possibilities, the number of things we can get, if you counted all these around the outside, you'll notice that there's 64 possibilities. There you go. And that's the basis of the genetic code. We have a certain number of amino acids, but there's only 64 real possibilities if you're using 4 different components in 3-letter words. So 4 nucleotides, 3-letter words, 64 possibilities. And I read just the other day that what researchers have been doing is they've introduced 2 new nucleotides, which is kind of mind-blowing. And they've increased the number of possibilities. Instead of 4 to the third, now we're looking at 6 to the third. And that's some interesting synthetic genetic life you're producing there. That gets into bioethics, but I think it's quite fascinating. So there's our codons that on the mRNA. We've got our anticodons on tRNA, and the stamping process is translation. So as these are put into place, we bring in the actors, the ribosome, which kind of looks like a hamburger with a big bun, little bun, and our amino acid delivery man, tRNA. And as this play works out, you're getting the stamping process. This is a picture from your book. I'm sorry about the graphic. I'm wondering where that black little piece of schmutz came up in my diagram. But this is a stamping process, and the play, the whole thing, is called translation. You have finally translated mRNA using tRNA into a protein. And that was the whole point. Protein production. And it's just one of those everyday miracles I talk to you about all the time. Here's a polysome, and you can see this happening if you have appropriate sort of electron microscope technology. You could see it happening in real time. How do we know? Well, here's an electron micrograph, and there is an mRNA strand right there. You'll see another one here. You have to know what you're looking at with these things. And another one here. So as these little guys are forming off to the side, that's the production of protein. So you can see this happening. What's a little bit hard to see is you're not seeing all the little ribosomes that have attached to this all the way along. But they have to be at the intersection points of these little squiggly primary proteins. these amino acid chains. So you can see it with electron micrograph technology. And there's a visual of it just below. So it proceeds really from 5' to a 3' end. So pretty neat stuff when you think about it. Now, this could be one of the greatest test questions. It just, okay, what's happening here? 120 marks. Well, what's happening in totality is that you've got your transcription of your DNA happening right up here. So I think I'd better, this is a picture, so I'll have to go to a different highlighter. There we go. So you've got transcription happening here. You've got your production of mRNA here. It gets cleaned up. You'll notice that the introns get sliced out, but the extrons remain. So you get your cleaned up mRNA strand, which then goes out to your ribosome, your rRNA. Look at all this RNA. Transfer RNA right here, our tRNAs, come in and stamp our amino acids, and here's our protein. If we go back to biology 11, you're thinking about this and you're saying, what would happen if there was a mutation to DNA? What if the master code changed and the nucleotide sequences were a little bit different? That, my friends, is a story for another lecture. But for now, we've come to the end of this one. So thanks for watching, and have a good one. Don't work too hard. Always remember, enjoy yourself. Science is fun.